Okay, the next macromolecule we're going to take a look at are proteins. And proteins have a lot of different functions. They're the most complex and versatile of our macromolecules. They're also the most common component of the cell. So you can see on this little graph over here that the most common macromolecule that our cells are made out of are proteins. So here are just some of the functions that the cells will use proteins for. So for example, one big function that proteins will do is they're used for cell signaling. So a lot of our hormones are made out of proteins. So for example, the hormone insulin, that's made out of hormones. So that will be released after you eat a meal and tell your body cells that, hey, there's a lot of glucose out there in your blood because you just ate. So it tells your cells to put out your glucose transporters to take the glucose into your cells so you can use that for energy or store it for later. And then a lot of the receptors that are on your cells that bind to the hormones or other messengers like neurotransmitters or things like that are made out of proteins as well. The contractile proteins that are in your skeletal muscles that allow you to contract your muscles, those are made out of proteins. We also have structural proteins like the collagen that is in our skin. That's kind of a rope-like protein that actually will strengthen our skin and makes it so our skin doesn't tear. A lot of the enzymes in our body, those are made out of proteins, and enzymes are used to speed up chemical reactions. Antibodies that are made by our immune system, those are also made out of proteins. And we can also use proteins to store amino acids. So for example, in eggs, the egg whites, those are actually a storage form of amino acids called ovalbumin. And those amino acids are there for the embryo to use as the embryo grows into the chick. And then we also have proteins that will be used for transport. So for example, in this picture, we've got a transport protein that's being used to transport things into the cell. So with the structure of proteins, the protein is the polymer, and the monomer that makes up the protein, the subunit or building block, would be the amino acid. And the bonds that join the amino acids together to make up the protein are called peptide bonds. Okay, so the monomers, the amino acids, here's a picture of an individual amino acid over here. There's actually 20 different amino acids that we can use to make up our proteins. And down here is just showing making a protein and what type of reaction would this be. So hopefully you recognize this as a dehydration synthesis reaction, so you can see that they're squeezing out the water molecule, dehydrating it to form the peptide bond to join the two amino acids together. Okay, and then there's 4 different levels of protein structure, and we just call them primary, secondary, tertiary, and quaternary structure. So I think it's just easier if we just take a look at a picture of this. So the primary protein structure, that's just the linear sequence of the amino acids. So what order the different amino acids are in. And then we'll take that linear strand of amino acids and then we start folding that. And we can form an alpha helix, which is just kind of like a coil. Or we can also form some beta pleated sheets. And that's kind of like an accordion fold. And then we can take those alpha helices and beta plated sheets and then fold those some more. And then the tertiary protein structure we get, this is just the overall three-dimensional shape of the protein. And then sometimes with our proteins, we might have more than one of these polypeptide chains joined together. So you can see a light gray and a dark gray polypeptide chain joined together, and that would be our quaternary protein structure. Okay, so if we have a change in our protein structure, that can really affect the shape of the protein, and if we change the shape of the protein, that can affect the function of the protein. So here's a genetic disorder where we've got a mutation in the gene that codes for a protein called hemoglobin. And hemoglobin is the protein inside of our red blood cells that carries oxygen. And so people with sickle cell anemia have a mutation in their DNA and the gene that codes for hemoglobin, where one of the amino acids has a change. So you can see that at position number 6 in the amino acids in the protein, we've changed that amino acid to a different one in sickle cell anemia. And because of that one amino acid that's been changed, is that we change the shape of the hemoglobin protein. So up here on top is what the normal shape of the hemoglobin protein should be. And down here on the bottom is the change in the shape because we've changed that one amino acid. And now the hemoglobin proteins end up sticking together when they should stay separate. And we end up with sickle cell anemia. And so the red blood cells end up with this sickle shape instead of being nice and round. And that causes sickle cell anemia. Okay, another change we can have in protein structure is called denaturation. So this is when the three-dimensional shape of the protein, we end up unfolding it. So this will happen if you heat up a protein or maybe change the pH around the protein, or certain chemicals can denature the protein. And when you denature the protein, when you unfold it, it loses its shape, and then the protein's no longer going to have its particular function anymore. A really good way to see a protein denature is when you cook an egg. So when you crack open the egg, put it in the pan, you can see the egg white is clear. But when you heat it up and cook the egg, the heat will denature the ovalbumin protein that's in the egg white, and that protein will denature, it unfolds and changes shape, and it turns white. And so that protein has now denatured. And so that's a good example of a protein denaturing, is when you cook an egg.